Welcome to a new webinar in the series covering innovative applications delivered to you by Waters. I'm Goro Bosch and will be your host. Today we are going to present the development of the UP-LC-MS/MS analysis for clinical research of thyroglobulin in serum using SYS-Kappa workflow. Our presenter is Dominic Foley. He has started his career at Waters in 2013 at our mass spectrometry global headquarters in Wilmslow in the UK. It's near Manchester. Dominic has been active in developing and validating bioanalytical methods following the CLSSI guidelines focusing on steroid hormones. More recently, he has been involved in the development of the quantification of biomolecules with LC-MS/MS, like monoclonal antibodies or thyroglobulin. Dominic has worked in separation sciences for 17 years, following his ambition of improving the quality of healthcare. Before we start our session, I will give you some information on how to use the platform. We encourage you to ask as many questions as you want. That is using the box on the left. So this Q&A, these questions will be answered during the Q&A that we'll have at the end of the webinar. Your opinion is very important to us, so please give us feedback on the event by answering the few questions that you can see on the right side of your screen. And finally, you have access to resources related to today's presentation by clicking on the links at the bottom of your screen. And without further ado, I will hand over to Dominic for his presentation on quantification of thyroglobulin in serum for clinical research using SYS-Kappa workflow combined with LC-MS/MS.
Okay, thanks, Goro. Welcome everyone to this session today. Good morning, good afternoon, and good evening. So today I'd just like to discuss some of the work we've been performing over the last year or so on quantifying thyroglobulin in serum and trying to get the sensitivity requirements we need using SYS-Kappa workflow and combining that with liquid chromatography with tandem mass spectrometry. So this is an outline of what I plan to discuss today. I know a few of you may not be totally up to date as to the nature of maybe protein or peptide-based LC-MS analysis. So I'll just do a bit of a recap around proteins themselves and the method development required for protein and peptide analysis by LC-MS. I will then cover the Waters workflow for the LC-MS analysis of serum thyroglobulin, followed by some key performance characteristics, and then finishing up with some summary and acknowledgements. So on to the protein structure recap. I know this is very basic for some, but I thought it'd be important just to cover this very briefly. There are 20 naturally occurring amino acids in the body, and these are the basic building blocks of the human proteome. These form the primary amino acid sequence and the polypeptide chain, which you can see on the bottom left-hand side. As part of this sequence, you have formation of peptide bonds. These peptide bonds, particularly when it comes to liquid chromatography and mass spectrometry, fragment in a very specific and predictable way, which is very useful. And also we can manipulate these particular amino acids, particularly in regards to arginines and lysines, when it comes to digesting proteins as well for their LC-MS analysis. So as I said, these specific amino acids do have a role, particularly role in protein folding. So cysteines can form disulfide bridges, and they also have threonines and serines that can form glycosylation linkages with glycans. Proteins fold in a specific way as well to perform a specific function, and this can be seen on the bottom part of the slide. So you have something that's as simple as angiotensin 2, which is only 8 amino acids long and a very small peptide, which is only 1 kilodalton in size. And then you have increasing structural complexity, moving through to the secondary folding of alpha helices and beta sheets, to tertiary formation of proteins through covalent bond interactions. And then you also can have quaternary folding interactions as well. So on the far right-hand side, you have a structure of what we deem a dimer of the thyroglobulin protein, which is over 600 kilodaltons in size. So a relatively large protein in the context of the human proteome. So in regards to the LC-MS analysis of proteins, there are perhaps two approaches you can take for this. You can either take the bottom-up approach, which we term the surrogate peptide workflow. And in this instance, you select a signature peptide, which is specific to the protein of interest. And this acts effectively as the surrogate for that protein. So this can be done using tandem mass spectrometry, where you effectively generate a tryptic peptide from digestion with an enzyme such as trypsin, and you can separate and detect that specific peptide. You could also take the top-down approach, where you can measure the intact protein using something like a time-of-flight system. Now, this can give you a lot of structural characterization information of the overall protein, but in context of what we're trying to do in this instance, absolute quantification isn't as good as, say, a bottom-up approach using a tryptic peptide. So there are benefits to this surrogate peptide approach, but one of them being that the compatibility of that approach fits into existing LC-MS workflows within a clinical laboratory. And this has comparable analytical sensitivity to a lot of existing methodologies, but with that brings improved selectivity, so the overall accuracy and trueness of the method should be much better. So on to some aspects of method development with the protein-based LC-MS analysis. So what we want to do is select a peptide from that protein. So what we want, first of all, you want to see if it's unique in terms of the human proteome. So what you would do is input your particular target peptides that you may have identified into a BLAST search online, and this effectively screens known sequences across the human proteome and also other species to see if that particular peptide is unique. And therefore, when you're looking to detect it, it provides you with accurate information. The peptide length can be quite important because that can have an impact on the physiochemical properties of the peptide. So you typically want this between 7 and 20 amino acids long. If it's too small in size, it isn't particularly specific. If it's too long, you tend to maybe have more issues around solubility or absorption in the system. So it's something that we try to keep track of in terms of a medium-sized tryptic peptide following digestion. Following this, you could also look at the digestion parameters. So avoid ragged ends. So these ragged ends are where in the sequence you could have a sequence of lysines or arginines, where you could have effectively miscleavage sites or less specificity of that peptide because of potential miscleavage or different areas of cleavage along that peptide. So you could lose sensitivity as a result of that. And miscleavage sites also where you have lysines and arginines adjacent to prolines. So in some instances, the trypsin or the trypsin enzyme may not necessarily cleave that particular site, or there could be a degree of inefficiency as a result of those prolines being adjacent to the lysines and arginines. And then you also want to consider reactive residues. So cysteines that can form obviously those disulfide bridges. So you can reduce an alkylate to help with those. Or methionines. So these are prone to oxidation. So the oxidation would effectively change the MRM that you're targeting for the precursor and product ion in the tandem mass spectrometer. You also want to try and avoid some modification motifs where possible. Basically avoid peptides near or peptides that do contain PTM sites, such as those that are prone to maybe phosphorylation or glycosylation, because those sites may impact, say, the capture in the sample preparation, and also, again, if you're targeting this by MRM, could change that MRM that you're trying to use for the analysis. And obviously, one of the key components is the actual observability by MS. So is it sensitive enough? Now, this can change between systems as well, so it's also important to keep an eye on that. But ultimately, that's the goal. If you're trying to demonstrate pure sensitivity, having a peptide that has particularly ionizable side chains will help. And arginines and lysines potentially could help with that as well, and any basic functional groups. And then the actual hydropathy, so the hydrophobic nature of the peptides, whether they're really hydrophobic, they could exhibit solubility issues. Hydrophilic peptides can exhibit LC retention time instability, so it alerts too early in your LC separation. Do you have stable retention time as a result of that? So in those instances, you may then have to play around with the mobile phase conditions to ensure that you have reproducible retention times. A lot of this can be done using something called the MassLink Skyline interface. So Skyline is a third-party piece of software that can link into MassLinks, which is the MassLink software, which is the Waters piece of software that runs our LC-MS systems. And this helps effectively perform a lot of this filtering process and helps you in terms of developing a method that you can test as part of a protein and peptide-based method development. And at the bottom of the page, there are a couple of maybe recommended reading in terms of publications that could be used for your own learning around protein and peptide-based analysis by mass spec. And the bottom right-hand side publication actually covers a lot of these aspects that I've talked about here. And then moving on from that, so you've developed your LC-MS method. You've got a strong understanding of what you need to target for a peptide. Now you want to try and develop some sort of sample preparation as part of that bottom-up MS approach. Now, there are some challenges around this. So there are different pathways you could take to get to the point where you've got enough sensitivity for this analysis. You could follow the pathway, perform a protein-level cleanup, which would maybe capture the protein and isolate it from other proteins in the matrix. You can then perform effectively a digestion on this and then do an analysis of the resulting peptide from that digestion. Alternatively, you could look at the denaturation, direct ingestion, direct digestion and injection, if you don't necessarily need the sensitivity. Or the other option then is to perform that digestion and then capture at the peptide level. Now, that particular approach can be quite specific because you're digesting everything in the sample and then only targeting your peptide of interest. So that's something that we look to employ with the thyroglobulin analysis, which I'll talk about later on in the slides. Now, there are some challenges around this. So what you could do here is you're evaluating different aspects of the method. So there's extensive method development usually involved in something like this, which requires a lot of expertise to a certain extent, particularly when it comes to like use of antibodies for capture of proteins and peptides. And a lot of these methods don't tend to be standardized. Typically with something like a protein or peptide-based analysis, if you are doing a capture in particular, and also the digestion, these historically have taken quite a long time. So people may do an overnight digestion. And those digestions as well can be quite variable, protein to protein. And digestion efficiency could also be an issue. So maybe taking that into consideration when you're developing a method like this. So I've got a poll question here just for a bit of audience interaction. And I was just wondering if you feel you have the technical expertise in your laboratory to develop large molecule LC-MS methods. And it's just a basic yes or no. This just gives us a bit of feedback as to whether or not we are able to offer any help in this regard, if our applications team can help with any training, if you are interested in these types of methods. So I'll just give you some time to vote on this, maybe another 10 seconds or so, and then I will move on to the results and the next part of the presentation. Okay, I'll finish that there. So, it's a fairly even split. So 45% do think they have the technical expertise and 55% no. So hopefully this webinar today helps a lot with those 55% in particular. So yeah, let's move on to the next slide. So sample preparation, as I've alluded to, could involve protein and peptide capture using antibodies. So this has risen in prominence over in more recent years. And immunoaffinity or immunoaffinity precipitation, or purification as they call it, can provide orthogonality to LC-MS. So what you may see is a rise in prominence for these types of methods. So not only thyroglobulin, but things like maybe insulin and the like. So we're targeting the protein or peptide, and doing this reduces the complexity of the sample quite significantly, which provides increasing selectivity. This does enable faster LC-MS separations. So for something that involves a direct digest, an injection, you may typically need quite a long LC-MS runtime to pull apart all the matrix interferences and potential ion suppression that could be impacting your analyte. This also has a benefit in terms of increasing the signal to noise. So a lot of these methods, particularly around amino affinity, have very low background noise, which helps with those signal to noise calculations. And the cleaner sample actually leads to reduced downtime on the back end. So as with most things with LCMS, if you do something like an SPE, it provides a much cleaner sample and gives you more predictability around instrument performance. The same is true here. So there are amino affinity isolation workflows, and I've briefly touched upon that in a previous slide, but effectively moving from top to bottom, you have a direct digest approach, where you just digest and inject and try and detect your peptide of interest that way. You have a protein-based capture approach. So you isolate the protein of interest, digest that, and then inject the resulting digestion. And I try and isolate the peptide that way. And then the peptide-based capture approach is shown at the bottom, where you perform the digestion and then the capture of the peptide. And more often than not, you get increasing selectivity, potential LCMS speed, and improved signal to noise moving from top to bottom with that approach. Some interesting reading around this was from a few years ago now, looking at the future state of these types of techniques. And it does cover a vast array of different analytes where you could potentially utilize immuno affinity with mass spectrometry. So onto thyroglobulin itself. It's, for those who don't know too much about thyroglobulin, it's a large dimeric glycoprotein. So two monomers joined together. And this is produced by the follicular cells of the thyroid. And it's used entirely within the thyroid gland, primarily to create T3 and T4. And the measurement of this has traditionally been performed using immunoassays. So the main issue with that is that antithyroglobulin antibodies, potentially heterophilic antibodies, depending on the type of immunoassay being used, can cause interference. And that can either have an impact in one or two ways. You could have non-determinate concentrations when you should expect to be seeing something, or higher than expected calculated concentrations as well. So the idea behind the LC-MS approach is that you can almost help circumvent this problem of that protein-to-protein binding type interference that can occur using sample digestion. So you effectively just digest the entire sample. This cleaves all the proteins. So the digestion reagent used is typically trypsin. And those proteins include the antithyroglobulin antibodies and also those heterophilic antibodies as well. So in this regard, from an analytical standpoint, it helps remove them as interferences. And there are some publications on this already. So using two specific tryptic peptides for thyroglobulin, shorthand the VIF peptide and FSP peptide, they have been shown to work quite well for this type of analysis. And a paper originally from Andy Hoofnagel from, I believe, 2008, first documented this type of approach, and improvements on this approach have been done since. And the idea as well, of the paper on the left-hand, bottom left-hand side of the screen, the idea was to try and harmonize some of these approaches where possible. And then leading from that, was the paper from 2022 on the bottom right-hand side, where you're using a distributable LC-MS method for the measurement of thyroglobulin. So the idea here was to try and generate like some sort of reference material that labs can use to help harmonize results between different laboratories. So moving on to what we have developed around the Waters workflow for this thyroglobulin method, we have the the sample preparation using the CISCAPA. So this is an immunoaffinity technique, which I will touch upon in a second, which helps simplify automation. It improves analytical sensitivity and maximizes the LC-MS throughput. We're automating the method as well to reduce hands-on time. And in this instance, we use the Andrew Plus pipetting robot. So we're able to fully automate the method on that system. This is quite affordable in context of automation platforms as well. And this, because of the lack of hands-on time, it's more simpler to use, can potentially help maximize the throughput a bit more. For the LC-MS analysis, we're using parallel column regeneration. This has two benefits. It helps improve the robustness by providing additional column cleaning. And also, because we're using two columns effectively in parallel, where you're maximizing the throughput in that sense. And then for the absolute sensitivity, which is required for this type of analysis, we're using a CEVO TQ Absolute MS. Now, this was launched on the IVD platform last year, and this is ideal in terms of overall footprint and performance for the method. And then finally, on data analysis, to help minimize touch time, data review time, and effectively faster turnaround time of results, I'm just going to show you some aspects of our relatively new software called Waters Connect for IVD with QuantReview. So CISCAPA. So this is a technique that's been around for around 15 years or so. So it's an immunoaffinity-based capture. CISCAPA stands for Stable Isotope Standards and Capture by Antipeptide Antibodies. A bit of a mouthful, but that's the approach we're using. It's a sample preparation method that improves the performance of mass spectrometry for measurement of pre-selected protein targets. So the peptide targets are captured, typically post-digestion, alongside a stable labeled internal standard. And this compensates for those changes in the capture recovery and any variability you may observe in the LC-MS analysis. So it helps effectively normalize the response. And the benefits of this CISCAPA workflow include some of the benefits I previously described. So eliminating the impact of those protein interactions. You can also analyze low abundant proteins. And then you have potential around something called stoichiometric flattening, where you may have something of low abundance and you want to analyze something with high abundance. So you can normalize those two analytes effectively into the same dynamic range by changing the capture characteristics for those analytes, so they can be analyzed in the same method. And as I touched upon previously, you have clean samples for rapid LC separation or direct analysis. So all this is done on like a magnetic bead-based platform. So this is an example of what you might see in the picture here. So the CISCAPA magnetic beads, when you're performing the capture and performing the washes and elutions, you pull the magnetic bead down into the corner of a plate using a magnetic array. And that allows you to remove your washes or your sample and remove them into waste while the analyte is captured on effectively what is that blob of magnetic particles in the corner of the plate. So I touched upon the simple affordable automation using this CISCAPA approach. This helps minimize the number of manual steps involved, controls that technician to a technician pipetting in precision, so it becomes more reproducible. There's less of a concern with user error. and it frees up that technician time for other tasks and minimizes those transcription errors. Now, for this approach, I said that we use the Andrew Plus pipetting robot with OneLab software. This is a low to medium throughput automation platform. It's positioned not in your more complex type workflows that more automated and flexible systems may be used, such as your TCAMs and Hamilton systems. And it's kind of in between those systems and something like an automated pipette, which is something like what Andrew Plus uses. They use the Pipette Plus type pipettes. So this kind of sits in between those types of systems. It uses a cloud-based software for the user interface called OneLab, which is a nice user interface and allows you to download methods from the cloud, and you can implement them directly on your system. And it provides you with a step-by-step walkthrough on how to set up and run your system if need be. We've also used the Hamilton Microlab Prep automated liquid handling system for this, just as an alternative device. This is probably in a similar price point to the Andrew Plus system, and effectively this helps automate the workflow in a similar manner. So more on the actual Andrew Plus pipetting robot with OneLab software. So we developed the protocol start to end, fully automated. So this was actually, there's an error in the slide there. It's not offline incubation, it is actually online, so it is fully automated. And all steps are performed across the deck. And for 96 samples, you can use all 11 domino positions. So a domino position is effectively a labware position. And within those dominoes, we have the Deepwell Magnet Plus, which is effectively your magnetic array, which can be activated and deactivated across the workflow to help perform your capture of your magnetic beads, to pull down your magnetic beads and capture your analyte. So this is just demonstrating the typical tip usage as well, and the consumables you may need to perform this workflow. You have the Andrew Plus pipetting robot overview here in OneLab. As I said, it's quite an intuitive interface. So on the top left-hand side, this is the screen you may see before you run any samples. So it's telling you what reagents are in what trough positions, what well plates you need, the type of internal standards you may require. And then the bottom right-hand side is what you may view when you're running your analysis. So it gives you a start and predicted end time. So what the expected runtime of the method is based on the number of samples you're running. It tells you what stage the method is up to as well, looking at the right-hand side. So it just gives you a rough idea of where the method currently is, and it allows you to plan your day around those steps. So in regards to that, I've covered a bit of automation there. We're just wondering what sort of sample preparation, if any, you have for automation in your LCMS lab. So I'll just give you maybe 10 to 15 seconds to answer this question. Just because there are multiple choices here, I may just give you a bit more time just to catch up. And I can have a quick drink. Okay, so we have some answers submitted from the attendees, so thanks for that. Okay, we have a bit of a spread, which is nice. So from an automation point of view, more people have Hamilton systems. but some people do have TCANs in the Andrew Plus, but 40% don't have any automation. Now, in regards to this approach, the method can be done manually, but it can take some time because you've got a number of wash steps that are involved in this process. And because with the pull down type approach with the magnetic beads, it's important that you are able to remove all the washes from every well, and then you have to do the eluent transfer at the end. So that can be quite involved from a pipetting point of view. So automation is generally recommended for this type of workflow, which is why we try to develop workflows around the more affordable automation systems. So moving on to parallel column regeneration, we have the thyroglobulin. So it's a single elution, it's thyroglobulin separation using a single elution pump and then a re-equilibration pump to do the regeneration step. So effectively, it's a single application running 2 columns in parallel, providing an increase in LCMS throughput. And then this is compatible with your existing software platforms. So it can do your LIMS export and data, LIMS import and data export. And it is compatible with liquid handling system method import as well. So the principle of this is shown on the bottom left-hand side, where you have the single channel approach, where everything is effectively run in sequence. And then in the bottom left-hand side, you have the parallel column regeneration approach, where you are doing a separation of an analyte, then equilibrating on that column. While that column is equilibrating, you've done another injection on the second column, which is separating during that equilibration step on the other column. So this happens in turn, in parallel across both columns. And by doing this, we're able to cut our run times from a single channel approach from 3.3 minutes down to 2.3 minutes for that column regeneration. And this gives us a 43% increase for the thyroglobulin analysis in terms of sample throughput. So we're up to around 26 samples an hour. So this doesn't necessarily have benefit just for thyroglobulin, but also just for general mass spec utilization. So if you have other methods that you're wanting to run on the system, you will also have time to run those as well on the same day because of that increase in throughput, which is quite nice. Moving on to the actual MS detection, we're looking to show in this slide the positioning of our TQ systems for endocrinology research applications and some of the minimum analytical sensitivity requirements for those. So for those who are on the call who are also interested in, say, steroid hormone analysis, this slide just provides a nice overview of what TQ platform you may want to choose for a particular type of analysis. In this, we also have the thyroglobulin. On the right-hand side, you can see there that it's underneath the TQ Absolute MS, and it will work perfectly well on TQ XS system as well for this application. We haven't tested on the Xevo TQS Micro for those who are interested in that. But this slide just effectively gives you an idea of what performance or what system you need in order to obtain the performance for the assay. So for those who aren't totally familiar with the Xevo TQ Absolute, this is a relatively new system by Waters. The idea is it's supposed to give you the best-in-class sensitivity in a smaller package, and also reduced operational costs. That could be associated with power and gas consumption, and also heating as well. So if you think about air conditioning demand, if the system's giving out less heat, then it's less of a demand on the air conditioning in your laboratory. The footprint as well, you can see in the diagram here, is generally a smaller footprint in terms of bench space compared to other vendors. And this allows you to make better use of the space in your lab, maybe fit more systems in the lab as well. So you're seeing that generally 50% reduction in demand for the power and the heating. So with that, you've got a reduction in footprint. So potentially you could fit another system in place in your laboratory without causing too much of an issue in regards to your facilities in the lab. And finally, in terms of the workflow, we have the Water2Connect for IVD software with QuanReview. And this is a new data review software package that improves usability and reduced data review time. Now here I'll just talk to show you a video of what this looks like in practice. So I'm keeping my fingers crossed that this now works when it loads. Okay, so apologies about that. The video wasn't playing. So the idea was I had some backup slides as well here, just in case there was an issue with the video. So on the previous slide, you can see the dashboard, which effectively is showing the overall view of how the batch has performed. So if you look at the dashboard here, you have a view of the standards, the performance of the calibration standards of the method, like what are any of the residuals not passing spec. You have the calibration performance, which is giving you the idea of the R² values. You've got the performance of the QC and the system suitabilities, if you are using system suitabilities. And then you have the internal standard performance. And moving to the right-hand side, you've got all the samples that are in your batch, and then exceptions or queries related to the injections in that batch. So there are different dashboards across the workflow. So in this instance here, you have the review of the CALs and QCs. And then what you have is the linearity. So the calibration linearity on the left-hand side and the associated residuals. And then on the right-hand side, you have the chromatogram associated with those samples. And that chromatogram can be changed in terms of the peak. So it can be manually reintegrated if necessary, or some of those QCs can be ignored effectively. So the Water2Connect for IVD software with QuanReview also has a peak integration page. So we have an overall view of the peak integration page. And this gives us a quick glimpse into how chromatograms or the samples have performed across the entire batch. Now, this can be made smaller or larger in terms of plot size, depending on your requirements. You could also change the exceptions in terms of the filters employed. And then looking at the sample types, you can filter on sample types. And this gives us an idea of how this is performing for different types of samples. So your standards, your QCs, your unknowns. So they can be filtered accordingly. And that's one of the main driving forces behind this QuanReview is the ability to review things quite quickly. So overall, this is the system solution in more detail. We have the Acuity UPLC R-Class Plus FL system. The two columns running in parallel, so looking at the HSST3 column in the column manager, the Xevo TQMS system, and the SysCapper magnetic beads for the capture. And then we have a 2.3 minute injection to injection time. This is how the workflow looks. So overall, going through the workflow, we're using 250 microliters of serum. We're performing a denaturation, adding the internal standard, and then digesting. We're capturing that peptide from the tryptic peptide, washing that, and then eluting it off the bead, and then injecting that sample onto the system. So the total prep time for 48 samples is around 3.5 hours. And because of that LC cycle time being around 2.3 minutes, the turnaround time for the first sample is around 4 hours. So with that in mind, I've just briefly discussed the workflow. And as you're on this webinar, I'll just be interested to know if you're looking to implement a thyroglobulin LC-MS method in the oil laboratory. So I have a poll question here, which should give us an idea of whether or not people are interested in doing this. So I may just give you 10 to 20 seconds to answer this. Just from the point of view of, it gives us an idea of when we develop these applications, are they beneficial to our customers? Okay, so short on the time, I believe, so we'll just quickly go through this. So maybe some people are just here for their educational requirements. So we have some people who aren't necessarily looking to implement this method, but yeah, it's good that maybe around 65% of people are interested in looking to run this very soon or when they have resource. So thanks for answering that. So very quickly, performance characteristics, the MS system check, and then system suitability. We have the system check to demonstrate that we have MS performance. So we've done this across 3 different systems and demonstrated we get the same performance. And then the system suitability checks the LC-MS with the MS. And that again has been looked at on two to three different systems before, and that provides us with a similar peak area shown here, around 12,500 for system suitability test injection. Analytical sensitivity and linearity, we're working down towards 0.1 nanograms per mil in human serum. So we're using surrogate serum for this analysis because of the endogenous levels typically found in human serum. But then we've also analyzed the human serum separately, and that gives us a similar sort of signal to noise performance. We've demonstrated functional sensitivity for this approach. So around 0.1 nanogram per mil is less than 20% CV. And we do get calibration linearity across the range. Now moving on to the performance characteristics around precision. So we're performing a single channel type approach, which is on the top table. And this was done with automated and manual approaches. And with these approaches, we're still obtaining CVs within 9.5%. And then we also looked at the approach with the column regeneration. And that CV is less than 10%. And it's important to note here, that's a CV across 2 columns because of the column regeneration approach we're using. So that's quite nice to see. And finally, we have some method agreements. So we looked at UK and EQUAS samples, 38 of them in total. And we did see for this some significant proportional method bias. And this was around 40%. But then when we did some calibration reassignment based on the EQA scheme, these then fell to within 5.5%. So this is potentially an issue around calibration more than method, the specific method performance. So in summary, this is the end of what I'm presenting in the webinar today. So we've demonstrated a method for thyroglobulin using LCMS for clinical research, and it helps improve the sensitivity, selectivity, usability, robustness, and sample throughput with the workflow we've employed. And then offline automation of the method can be adopted, which helps improve adoption of the method and reducing the touch time and effectively overall laboratory efficiency. And we get greater performance across the range in terms of precision, linearity, etc. And then touching upon that last performance slide, standardization of the method is key in obtaining accurate results. And if you can't standardize, maybe harmonize. So there's some acknowledgements here. So some people across different laboratories in Birmingham and down in London at St Thomas's Hospital. We've got Morty at SysCapa for helping with this method. And then we've got some internal folks at Waters, Rob, Mary, and Nadim, who helped with method setup and the automation of the method. So at that moment, I could pass to Gordo for maybe 5 to 10 minutes of Q&A. Thank you.
Thanks a lot, Dominic. Yeah, while you were presenting, we've had a few questions coming in, and you can still type your questions in the chat, in the Q&A box. So please do so. So here we have one which is about Skyline. So I'm not familiar with the Skyline interface of MassLynx. It searches the best MRM transitions. That's a question. Or is a workflow to follow?
Yeah, so Skyline is effectively like a workflow. So you set up within Skyline some default methods. And when you are interested in a protein of interest, you use Skyline to develop some targets. And then the MassLynx interface allows you to basically optimize those targets and the performance of those targets within MassLynx. So it's kind of like a back and forth type optimization scenario. So that this is available as part of the MassLynx packages. So you could effectively, if you are interested in employing this, it's something you can look to evaluate.
Okay, sounds good. Why was SysCapa selected as the immunoaffinity approach? Have any other immunoaffinity-based approaches been tested in this workflow?
Yeah, so primarily this was a bit of a collaboration with SysCapa. So they have developed this antibody, the monoclonal antibody on a magnetic bead. And they wanted to just raise awareness of the value of thyroglobulin applications by LCMS, and we agree with that. There are other avenues you could explore, potentially around peptide-based capture. But I don't believe, unless I'm mistaken, something that's commercially available and been thoroughly demonstrated in literature. You could potentially do a protein-based capture, but there are potential issues around that in regards to those protein-protein interactions that I was discussing that could impact that capture of thyroglobulin. So realistically speaking, a peptide-based capture is preferred for this type of approach, which is why we used it.
Excellent. Another one related to SysCapa. So what is the commercial source of the SysCapa antibodies or antibody-coated particles?
So if people are interested in this, I would, you can purchase these antibody-coated particles directly from SysCapa. So you can go to their website and contact them through their website. And generally, they're quite good at getting back to you with a response. And they will provide a tailored quote for yourself. in terms of any evaluation you may be interested in doing for this workflow.
Maybe a last question. What type of internal standard was used for this analysis? Is it expensive to purchase?
So the internal standard is an interesting one. So historically speaking, there was a protein labeled internal standard, and that's generally ideal for a protein-based analysis, particularly if you're doing a surrogate peptide approach. But that was only very briefly available. So an alternative approach would then just use a isotopically labeled internal standard, or even a winged peptide internal standard. So effectively, this is an internal standard that has cleavable sequences, so like lysine and arginine, either side of the peptide of interest, and that helps compensate for digestion. Now, we didn't use that, but that's something we're looking to maybe evaluate in future studies, just to perform that comparison and see what the difference is between a winged peptide and a stable labeled standard peptide for some types of samples.
Okay, well, thank you. Yeah, I think we are a bit over time. Very interesting talk. So if you have further questions, do not hesitate to send them over to us. Well, you still have access to the collateral that you can download from this page that you are looking at the bottom. Thank you, Dominic, and we'll see everybody at the next webinar that we'll set up.
Thanks, everyone. Have a good day.
Thank you. Bye-bye.